Good morning, everyone. My name is Richmond Sarpong, and I'm an associate professor in the Department of Chemistry in the College of Chemistry here at UC Berkeley. And it's a pleasure for me today to introduce one of my colleagues, Professor John Hartwig, who's the Henry Rappaport Chair for Chemistry at UC Berkeley. But before I introduce John, I thought I would set the stage a little bit and put things in context. So I'm an associate director in the Berkeley Center for Green Chemistry for New Chemistries. And obviously, chemistry is an integral part of the green movement, as we all know. And chemicals are an integral part of this as well. There are good chemicals and bad chemicals. We tend to focus on the good chemicals and good practices for how to put these good chemicals to use. And I can think of no better person that is really practicing the different innovations of how to put good chemicals to use than Professor John Hartwig. So John has worked in a variety of areas and is a leader in the field in a lot of the areas that were mentioned here as the focus for principles of green chemistry, including catalysis, chemical synthesis, atom economy. I can go on with this list. But I'll mention just one of the things that John has really been a leader in, and this is this area of cross-coupling reactions. And so these are reactions where you can bring together two chemical entities to join them, and this has been a very important area for building pharmaceuticals, for example, and new materials. And in fact, this is an area that was awarded the Nobel Prize in 2010. And I sincerely believe that this is not going to be the last time that a Nobel Prize will be awarded for an area that's been so important to chemical synthesis. In addition, John works on the area of CH functionalization. So one thing that I will say is that for chemicals, a lot of our chemicals come from petroleum. And from petroleum, you obtain a lot of compounds that contain carbon-hydrogen bonds. So that's maybe a little bit esoteric, but we're looking to make use of these carbon-hydrogen bonds in building useful chemicals. And John is really one of the pioneers and leaders in this area. He has garnered far too many awards to mention here. He tells me that I need to keep my introduction short to give him as much time to spend really sharing with us some really exciting areas on catalysis for synthetic efficiency. But I'll only mention that just this week he's been elected to the National Academy of Sciences, and so clearly his contributions have been well recognized. So without much further ado, I would like to congratulate and welcome John to the stage.
Okay, well, thanks very much for the introduction, Richmond, and for the invitation. It's really an honor to come to speak at this symposium, to this broad audience, to a symposium which has a very broad range of speakers at a very high level. So when I got an invitation to speak here, I thought that was sort of an interesting choice because if you do a keyword search for green chemistry, you will not find us in that search. I think there's a paragraph of a paper where you have a red copper compound that turned green, and so if you search green, maybe you find us through that one paragraph. But as Richmond said, my group works in the area of discovery and development of reactions that are catalyzed by transition metal complexes. And over the years, as a community, we've developed a large body of reactions which has really changed the way chemical synthesis is done. And the principles of green chemistry have really become just part of the everyday mindset of people working in this field. So when asked to speak here, I was sort of forced to think about what which of those principles we do think about on an everyday basis and how to put together a seminar that would highlight some of those aspects and how we as a community do catalysis. So this will not be a sort of straightforward, straight-ahead research talk. I'll really spend about half of the time talking about some of our own research and try to highlight some of the aspects of our research that apply to some of those principles. And about half of it will be on background of the field and what I think people have contributed in the field of catalysis to the topic of today. So the general theme, though, is the title shows, is synthetic efficiency, really minimizing the number of steps and lowering temperatures, increasing yields, increasing selectivity. Efficiency can be defined many different ways. So what I want to talk about today is outlined here. So I want to give an introduction to catalysis. I talked to Marty before the talk began as to how much chemistry people have here. So forgive me if I start at too basic a level, but I wanted to sort of start at the very beginnings of catalysis. So I'll talk a little bit about catalysis and how that relates to green chemistry. Give you some examples of work that's been done in the field on processes that have come online recently with the kinds of catalysts that we work with, but not our catalysts, in response to a desire for greener processes. And then I'll talk about some of our own work, talk about a little bit about cross-coupling and its effect on how synthesis of certain classes of molecules are done, and then talk about CH bond activation and functionalization and how that also changes the way one can think about doing chemical synthesis. All right, so for the very basics, what is a catalyst? I'm going to be talking about catalysts all through the talk. And so the definition over 100 years or more years old is that it's a reaction component that one adds that doesn't change, but does change the way a reaction occurs. It makes the reaction occur with a faster rate, and it can affect selectivities, as I'll talk about. So catalysts are everywhere, whether they're in your own body as an enzyme that does chemical synthesis in your own body or in any organism, whether it's the smallest possible catalyst, just a proton, or whether it's a catalyst that's often used on very large scales to make chemicals in billions of pound per year scale that would be a heterogeneous catalysis. Or the kinds of catalysts that I'll focus on in most of my talk today, which are called homogeneous catalysts. Homogeneous because they're in the same phase as the reactants that's all dissolved in a solvent, and they contact each other that way. And so catalysts will take reactions that seem like they're impossible. They'll take two sort of incompatible reagents and allow them to come together to do a reaction in a way that would be selective and make a desired product, taking things that would take a geologic time scale or longer, able to be conducted in an afternoon in a laboratory. So they make those reactions more efficient, again, by lowering temperature, increasing rate, increasing yields. But also, one thing I want to highlight today is they can change the selectivity of a reaction because selectivity is dictated by how fast one reaction occurs versus another reaction, and a catalyst can make this one faster and so lead to that product rather than leading to another one. So how a catalyst works and what it does and does not do, right? It can't change the thermodynamics for a reaction. So it can't make something that's unfavorable become favorable in terms of its free energy or enthalpy or entropy. So you can't take a catalyst, drop it in, and all of a sudden split water and make hydrogen and oxygen without some input of light, for example, to make that happen. So the way a catalyst works is by lowering the barrier for a reaction, depicted by this here. And it does so by changing the mechanism of the reaction. So it now occurs, instead of in one step, it would occur here, as I just schematically showed, in 3 different steps, each of those with a lower barrier than the first one. And so the challenge in some of our research is to understand what those different steps are, what the barriers are, and what the relative energies are. And the creative aspects of developing a catalyst is to imagine what those steps could possibly be to make the reaction happen. Okay, so the one last point a reminder for myself on this is to say that one of the things that the catalysts allow us to do is to take less reactive molecules and put them together because it lowers the barrier. And typically, a less reactive molecule is something closer to those petroleum feedstocks or biofeedstocks that Richmond had talked about. So when we do reactions on molecules, often we will increase the reactivity of those molecules by installing what we call functional groups, right? Places that reactions occur. Let's see. You can plug in one end of but you have to plug in both ends. There we are.
That's yours, right?
Yes, that's my fault. Yeah, we're good.
Okay.
No problem. All right, so one of the metrics for how good a catalyst is how long it lives and how much product it makes for the amount of catalyst that's added. That metric is called turnover numbers, and so I'll refer to that a couple of times here. Just to put that into some perspective, if a catalyst is good, at least for fine chemical synthesis, you get on the order of 1,000 or more, better 10,000 turnovers. And so this would be the catalyst. These are 2 aspirin tablets. And then here's a 20-pound bag of rice. So from this amount of catalyst, you would make that amount of product. To put that into some perspective, that bag of rice, at least by our family, cannot be consumed before those little thin moths come over and take over and start eating it all and overtaking our cabinet. All right. OK, so catalysis in industry is done on many different scales. And together, that leads to a large scale. And so by some estimates, it's trillions of dollars or a good fraction of the US GNP. And so how can that be? Well, that can be because it's done on scales that are very large for petroleum processing, all the way down to small scales to make very high-value products. Some of them were referred to in the previous talk. OK, just to give a couple examples. So one of the favorite examples, at least for introductory chemistry and even in current research, is ammonia. Right, so ammonia is made on an enormous scale by the reaction of nitrogen with hydrogen. So just 2 elements, really, coming together, no side products, you make ammonia. What could be cleaner? Well, this is done on the scale of about 200 billion pounds per year, the numbers from 2004. And it's, from this website at least, it stated that the growth of the human population was a function of the discovery of the Haber-Bosch process, this reaction that makes ammonia. And so a few estimates is that half of the nitrogen in your body, so the nitrogen in your nucleic acids, the nitrogen in your proteins, come from this chemical reaction. It can be traced back to that reaction. And another estimate is that about half of the world's population then could be only about, or more than half of the world's population requires the Haber-Bosch process. So without that process, we'd be from 7 billion, of the 7 billion people, we could feed about 3 billion. And if the population's going up to 10 billion, then we could feed only about 1/3 of the world's population without that reaction. OK, so that looks straightforward, except that now where does the hydrogen come from for this reaction? Well, that hydrogen comes from a petroleum reforming. So from methane reforming, we get hydrogen for that reaction. So although there's no petroleum seeming to appear here, that's actually where that hydrogen comes from. And so if we think that in the future we're going to get hydrogen from water splitting, okay, maybe we can make some hydrogen that way, but the pressure of hydrogen in this reaction is hundreds of atmospheres. And so we'd have to be able to split water in an efficient enough way to build up pressures of hundreds of atmospheres to run that reaction. So that's a huge challenge that is, sort of hard to imagine achieving. So that's one little snapshot of very large-scale catalysis and what some of the issues are, hidden issues are in terms of green chemistry, like where is that hydrogen going to come from that we would use in that equation to feed all these people. All right, so I assume this will appear in almost every talk today, but I thought I would go through some of the principles of green chemistry and how catalysis relates to that. So of the 12 principles of green chemistry, catalysis is number 9. Maybe it'd be nice if it was higher in that list and not just number 9. But the point I want to make today, really, is that catalysis might be number 9, but catalysts are needed for all of these, from 1 through 8, and probably even 10 through 12. Okay? And so, for example, catalysts, right, they create reactivity from less reactive compounds. So in the context here, that might, in many cases, means those compounds are less hazardous. It reduces the energy consumption, temperature, but also pressure. Right? Anytime you compress gases, that costs a lot of energy. They match and create compatibility of functional groups. And so this auxiliaries here often refers to putting something on a molecule to make that group no longer reactive in certain parts of the synthetic sequence. So that allows for greater compatibility of functional groups, removing those auxiliaries, allows use of new feedstocks, and creates rapid access to new classes of molecules. So some of these designed for degradation principles here. So maybe catalysis should actually be up on the top here and should be with a colon, and then all of these things follow from catalysis, right? So I don't really say that facetiously. I say that with some seriousness. Okay, so the thesis today, though, is that catalysis can alter selectivity. And I want to emphasize how selectivity and reduction of steps really just in itself leads to a greener synthesis. And so let me give a couple examples to start, not of our own work, but of things that have been done with homogeneous catalysts in industrial processes from large to small scale. So let's start with maybe smaller scale. So this is Crixivan and Dinavir, a compound that was developed at Merck. And it was one of the first, it's no longer one of the most active, but it was one of the first compounds used for treatment of HIV/AIDS. And so this is a molecule that is pretty complex in structure, even for a pharmaceutical, and it has 1, 2, 3, 4, 5, 6, 5 stereocenters in it. And so one of those is generated through a homogeneous catalytic process with a rhodium complex with a chiral ligand. I don't show the exact one here. So that double bond gets reduced to a single bond, setting that stereocenter. And so you could think of various ways to make that compound, but without a catalytic asymmetric process to set that stereocenter, you would throw away half of all of the product. And so all the materials that are used to get to that compound, you would be throwing away half of those all the way up through. And so just this simple catalytic asymmetric hydrogenation of a double bond led to a much cleaner synthesis of this compound. And I think the synthesis of this actually won one of the early green chemistry awards. So a second example is one of Richmond's examples from the introduction of cross-coupling, a Negishi cross-coupling. And Negishi was one of the recipients of the Nobel Prize for this chemistry that makes a carbon-carbon bond between biaryl groups. And so one of the messages here is that the ability for us to, as chemists, even make compounds that have certain activities relies on the types of reactions that are available. So this is Losartan. There's a whole series of sartan drugs that are antihypertensive, blood pressure lowering compounds. And so the way this compound is made in the first step is actually pretty straightforward now, using a Negishi cross-coupling reaction to make that carbon-carbon bond. But if you thought of another way to make that molecule, if you were not able to make that carbon-carbon bond, then you would need to be able to install that cyano group or this methyl group within this biophenyl framework, and there aren't very good ways to do that. That'll be the subject of the second part of the talk. So from here on, it's not the greenest synthesis, but it's pretty straightforward to get to that final product. Okay, a third example is now sort of increasing scale, now going to agrochemical products, things that one just distributes out into the field. I moved here from Illinois, and we saw lots of cornfields and lots of herbicides applied to those fields. And so one of those compounds is called dual Magnum, and one of the products is called dual Magnum, and that's the molecule, actually the active form of that molecule. Again, it's chiral. It's got a stereocenter and the position on the carbon alpha to nitrogen. This is the inactive enantiomer, the other hand of that compound. So this is the active. So these can be traced back to this one, which is the active enantiomer. And so you want to make that compound and not that compound. And so this was done, again, by an asymmetric hydrogenation. It's actually the largest scale asymmetric hydrogenation reaction that's practiced in industry. And it's not a spectacular reaction in terms of selectivity. There's about a 9-to-1 ratio of one of the enantiomers over the other. But the benefit from this, and why this compound is made in enantioenriched form, is that it's done on very large scale, so thousands of 10,000 tons per year. And all of that material then gets applied to the field. And if you're applying a 1-to-1 mixture of those two compounds, you need to apply twice as much than if you were applying a single enantiomer of that compound. It's not a single enantiomer, but it got cut off, but it's about 40% less that one needs to apply to the field instead of 50% less. Okay? And so actually this process really led to this whole family of phosphines that are bound to iridium, and those ligands are used for many other catalytic processes, including those that are made to make pharmaceuticals. So my last example from industrial processes is a recent synthesis of methyl methacrylate. Methyl methacrylate is the monomer that's used to make lucite, that clear plastic, right, that we use everywhere. And so the classic synthesis of this comes from acetone and hydrogen cyanide, one of the most toxic compounds out there. So you make acetone cyanohydrin, and then in sulfuric acid, you do a hydrolysis of the nitrile to the amide, make a sulfonate, then an elimination reaction, you get to methyl methacrylate. It doesn't look like such a bad synthesis in terms of steps and so forth, but you get a stoichiometric amount of this ammonium sulfate byproduct. And so if you consider that there are, again, millions of tons or billions of pounds of methyl methacrylate made per year, that's millions of pounds or millions of tons or billions of pounds of ammonium sulfate that is also generated as co-product that needs to go somewhere. Right? And we don't want ammonium sulfate for fertilizer. We want ammonium phosphate for fertilizer. So we need is there a better way to make this compound? We need a new synthesis. And so this synthesis came online fairly recently because of the development of a combination of a palladium catalyst, BD's palladium, and a particular ligand. And I won't go through all the development of that ligand here. But there's a new type of ligand that was developed that would bind to palladium and allow people to put ethylene, carbon monoxide, and methanol together. They all just come together in a single reaction to make this methyl propionate. So then you take this with formaldehyde, condense that in just sort of soft elementary organic chemistry to generate methyl methacrylate, and then the only byproduct that's produced in this process is water instead of the ammonium sulfate byproduct. And so this is the monomer to make the product that we often call lucite. It's the lucite process. And so this was also, I think, a process that was a recipient of a Green Chemistry Award. All right, so then from my group, we've developed a number of reactions that are often used now in various contexts in organic synthesis, not on the scale of the lucite process that makes billions of pounds per year. But what I want to use is these as examples to show how we, as a community, think about doing chemical synthesis and how we think about efficiency and selectivity having an impact on how that is done. And so one of these reactions is the coupling of an amine with an aryl halide. One is the coupling of a carbonyl compound. One is the cleavage, a selective cleavage of a CH bond of an aromatic compound. And all of these are enabled by some of the fundamental organometallic chemistry that's done in my lab. So let's start by talking about the synthesis of aromatic amines. So a very large fraction of pharmaceuticals contain aromatic amines. Something like 30% of them or maybe more contain aromatic amines. Polyaniline is a conductive polymer that's made up of just aromatic amines. And the organic layer, the whole transport layer of organic light-emitting diodes are also made from aromatic amines. And so if you go to a sophomore organic chemistry textbook and look at how you make aromatic amines, you make them starting from an arene like benzene. And you do a nitration with nitric acid and sulfuric acid, and then you would do a reduction of that, and you might use hydrogen, but in other cases you might use some main group metal hydride reagent, which leads to byproducts. And then if you just want to deliver one alkyl group to that, at least when I took sophomore organic chemistry, they taught us that you put a blocking group on there first, so that you don't put two of those groups on, you put only one. So you put the blocking group on, you alkylate, and then you take the blocking group off. So just to go from here to make an aromatic amine with one aryl group and one alkyl group would take these 5 steps. Each one requiring a separation, crystallization that involves solvents, putting groups on, taking groups off. All right, so could catalysis provide a more efficient way to make those compounds? And so neither one of these steps individually is particularly green, right? The first one is a chlorination of an aromatic to make an aryl chloride, and then the second step is chemistry that we helped develop to make the carbon-nitrogen bond, and so you make a halogen stoichiometric halogen byproduct there. But despite the fact that you would look at neither one of those individual steps and think of it being particularly green, this is a 2-step process to go from here to here instead of a 5-step process. And then one reason why this has become so often used is that there are about 20,000 of these aryl chlorides that are available, so most chemists then would just need to run this one reaction to couple the aryl chlorides or the aryl bromides with an aromatic amine. All right, so let me give you some examples of those reactions, and just to give you one slide to illustrate what it took to get to that point. All right, so we've worked on this, and other people in the community have worked on this for a number of years. And so the very first processes were reactions of the aryl bromides with amines or alcohols or thiols with a palladium catalyst and a base to make the carbon heteroatom bond in the product. And Steve Buchwald's group at MIT and my group when I was at Yale first published those about the same time, and we've been working on those processes since then. And so we've gone through a first-generation catalyst to a second-generation catalyst that had a chelating ligand. For those who are here are chemists and inorganic chemists, a ligand with 2 donors bound to the metal. And then we all of us switch to ligands that are very large alkyl substituents and have only 1 phosphorus bound to the metal. And then in our 4th generation version of this, we combine the properties of the 2nd and the 3rd generation to have these very sterically hindered alkyl groups at phosphorus, but also in 2 of them, so we have a chelating ligand that makes the catalyst stable. And so the latest results here, not exactly the absolute latest, but some of the things that we can do with this latest catalyst that we've worked with is to combine that with palladium to take an aryl chloride with a simple alkylamine, and just to show a very simple case here, to form this product now at room temperature instead of elevated temperatures and high yield. But maybe more important is that because of the combination of stability of the catalyst, but also very high reactivity, we can combine those two to make the carbon-nitrogen bond and this alkyl arylamine product now with about 20,000 turnovers. Right? So we can now got to the point where you can take these aspirin, sort of sized amounts of catalyst and make big bags of product. And so this is now reasonable to be conducted and as a commercial process, at least in the context of agrochemicals and pharmaceuticals. So that's one version of this reaction. But while we were developing that reaction, many people said, well, can you just make a primary arylamine? So can you couple an aryl halide with ammonia? And at least some aromatic amines, anilines, are made by taking phenyl chloride with ammonia and base and high pressures and temperatures. And so we always thought, well, no, we can't do that, but we can almost do that. We can do this sort of the equivalent of that by, say, starting with benzophenone imine and making that CN bond, and then we would hydrolyze that off to make the primary amine. Or we take this hexamethyl disilazide that's typically used as a base. We were happy that we could then use that as a nucleophile and then just hydrolyze that to make the primary amine, but you've got two equivalents of the silane product you get out. And then we can improve the functional group compatibility of this by now adding zinc to that reagent, to this reagent. And so these were reactions I think were synthetically valuable, and I think some people still use those reactions. But it turns out one objective that would be important would be to use ammonia itself to do that substitution. That would get rid of all those blocking groups and auxiliaries that are in one of the principles of green chemistry that we'd like to eliminate. And so with that catalyst I just showed you, we could do that. And so what Yang Vo, a graduate student in the lab, showed is that we could use just a small amount of excess ammonia with an aryl halide with that ligand and a particular palladium precursor. You've got to get that right to make this work, but then that would work to give a variety of different aromatic amines, now without any blocking groups, going straight from the aryl halide to the primary aromatic amine. Okay, so what that chemistry and the Negishi chemistry and the chemistry that many others have developed has shown is that one can start with an aromatic halide and then do, quote, cross-coupling chemistry to generate this whole series of products. And so this is important for medicinal chemists because they can start with this one material and they can generate a whole series of different products with different R groups, different substituents all around, and look at the SAR, the structure-activity relationships of those compounds, and use that to develop a drug. The same is true in the agrochemical industry. But a second question is whether one can do the same kinds of reactions, but where one would cleave a carbon-hydrogen bond instead of a carbon-halogen bond. And if one could do that, then you wouldn't need to necessarily install the halogen. You wouldn't need to do the chlorination step first before you did the introduction of all of these substituents on that aromatic ring. Okay. Maybe another viewpoint is that one could just do these reactions here, leaving the halogen behind, and then you could conduct all these reactions at the aromatic halide, and then you could increase the diversity of compounds that one would make with these aromatic derivatives. And again, this is important for the compounds that have biological activity. Agrochemicals and pharmaceuticals, aromatic units are sort of the bedrock of all of the compounds that are discovered in those areas, and also for material science, where the pi systems of those compounds are important for conductivity and hole transport and redox properties and so forth. Okay, so that question then leads me to the question about leads me to an introduction to carbon-hydrogen bond activation and what some of the challenges are to selectively cleave a carbon-hydrogen bond. So first is a problem of thermodynamics. And so these would all be great reactions to do, particularly this one down here, to take an alkane plus a water, make hydrogen and an alcohol that would be one of the commodity chemicals that's out there. But the problem is that all of those reactions are endothermic. So if we add a catalyst, we can make the reaction faster, but it's not going to go anywhere because it's uphill to make those products. And so we can do dehydrogenation, but you need to do something else to consume that hydrogen, and once you add that other thing, then you don't make that reaction practical anymore. Okay, so that's one problem. The second problem is selectivity, and so as Richmond pointed out in his introductory comments, there are lots of carbon-hydrogen bonds around molecules, and so selectivity is really one of the big issues. So if we have a molecule with different types of carbon-hydrogen bonds, then how do we get selectivity for one over the other? Radicals usually are selective for the weaker bonds, and this secondary CH bond is weaker than the primary CH bond. And so you would get a secondary alcohol. But then the problem here is that the products are more reactive than the starting materials, so this goes on to make other things all the way, when you burn it, to carbon dioxide and water, here to polyhalogenated products. So is there a way that one can use a catalyst and then make the strongest CH bond be the one that gets cleaved and then introduce some functional group at that position? So that's been one of the real challenges, is to be able to do this on aliphatic systems, so compounds with no pi system in them. But maybe an equal or maybe even harder challenge is to do that on an aromatic system. And so there are a variety of different ways that one can do selective transformations of carbon-hydrogen bonds, whether it's using an alkyl lithium with a directing group or taking advantage of the principles, again, of introductory organic chemistry, where if you have an electron donating group, the reactions go ortho and para to this position and that position. If that's an electron withdrawing group, the reactions go there. But what if you have an electron withdrawing group there and you don't want the substituent to be introduced here, you want it to be introduced there or there? Is there some other way some way to use a catalyst to change the selectivity of those reactions? So that brings me to compounds with metal-boron bonds. There's a solution to that problem, and there was by no means, that we looked at these compounds in the first place because we had all this vision about how to change, the selectivities of aromatic CH bond activation. But trust me, I'll get to that in a minute. Okay, so the compounds that we began to study very early in my career had metal-boron bonds, and we were interested in the properties of these compounds where you would have an empty orbital at boron, a p orbital that could overlap with the d electrons at the metal center, making a pi bond, and how would that affect reactivity, and how would the latent Lewis acidity of the boron perhaps play a role in selectivities. So what I'll talk about is how those properties led us to discover a CH bond functionalization reaction using boron reagents, how that led to a platform for practical CH bond functionalization, and how we've been able to use some mechanistic data to develop some new transformations. And all of this is in the context of selectivity and more efficient synthetic sequences, cutting the number of steps in a synthetic sequence. So the very first observation was a bit serendipitous. Under photochemical conditions, if you had light, the carbon monoxide ligands come off the metal center, leaving an open site where the CH bond cleavage can occur to make these compounds with an aryl boron bond. And these are very well-known synthetic intermediates that have been used in the Suzuki cross-coupling chemistry that I talked about earlier, as well as just oxidations to make phenols. But this is far from chemistry anyone would practice. Now, one of the early observations we made was the same reaction could be done on an alkane, and the reaction would occur at that stronger primary CH bond over the secondary CH bond. But these were not catalytic reactions. These were stoichiometric reactions. So we needed to convert that to a catalytic process. And the way that we did that was to turn to these reagents, which became commercially available about the time that we were thinking about this chemistry. And so what was known from some of the early reactions of these compounds is that a metal would insert into the BB bond. That would make a compound with two metal-boron bonds, which looks a lot like the compounds I showed on the previous slide. And so if those would also cleave a CH bond, then maybe you would make two products, the borane and the functionalized product, leaving behind this metal fragment that would insert into the BB bond to regenerate the starting species, and we'd have a catalytic cycle. And that worked. And so if we switched to a rhodium complex, which instead of having carbon monoxide bound to it had an arene that would dissociate from the metal center, then this rhodium compound would insert into the BB bond to make a compound with metal-boron bonds that would serve as a catalyst. And so in some early results, we showed that alkanes would react with these diboron compounds with this rhodium complex now as a catalyst to place this boryl group at the end of the alkyl chain. And with this group here, now one could oxidize it to an alcohol or do some cross-coupling chemistry. So that was some of the early results. Very far from anything someone would really use as a synthetic method, but was a starting point. So that project took a bit of a turn with a collaboration with a visiting scientist in my group, Tatsuo Ishiyama, who was in Hokkaido. Hokkaido is where the Suzuki-Miyaura reaction was invented, and he was an assistant professor with Miyaura. And then, as I said, was a visiting scientist with me. And what we found was that we could develop a related reaction with arenes instead of alkanes, using the same diboron compounds with a closely related metal. Iridium is below rhodium in the periodic table, by slightly changing the ligand. And so in some early results, we found that Tatsuo actually found that with bipyridine bound to iridium that you could make this product, but not very reliably. And the reason why you couldn't do it very reliably is because there's a long induction period. And so we began to do some mechanistic studies on this in my group. And what we found is that the reaction goes along and does nothing for a very long period of time. And so if you check the reaction here, you're disappointed. But if you wait just 8 more minutes and checked it here, you get almost quantitative yield and a great reaction. Right? And so then it was our job, as people interested in the mechanisms of reactions, to figure out what's going on in this time period, how to get rid of that induction period so the reaction is all going on here, and it would be a fast process, reliable, and then maybe we could do the reaction in a more practical way where we would not need a large excess of the arene. So I won't go into the details of that. We did sort out what some of the effects are going on in the induction period. So this is iridium with cyclooctadiene. I meant to put that abbreviation in the slide. Cyclooctadiene gets reduced. You get some pinnacle borane from the diboron compound. Just trust me, we sort of sorted out what goes on in that induction period here. And then we could figure out how to change the catalyst so that that induction period was shortened without having to do a lot of extra stuff. And so what we did, actually Tatsuo changed the chloride to a methoxide. We changed the bipyridine to ditert-butyl bipyridine. And you put those together, and you have a reaction that occurs now at room temperature with a 1-to-1 ratio of the boron to the arene, not an excess of an arene. And the reason why this reaction has become a synthetic method, and something Richmond's group has also used for some complex molecule synthesis, is because of the selectivities. So the selectivities of these reactions are controlled by steric effects, how big substituents are around the ring. It is not controlled by the electronic effects of sophomore organic chemistry. And so if you just look at the molecule and which CH bond is most available, for symmetric cases, there's going to be 1,2 disubstituted arenes. There's a single product. For 1,4, there's a single product if it's symmetrically substituted. But if you have 1,3 substituents on an arene, there's one CH bond that's different from the more sterically accessible than the others. And so that's the product you get. And because of the symmetry, it doesn't matter if those are the same or different substituents, you get a single product. And it doesn't matter if they donate electron density or if they withdraw electron density. What only matters for arenes is how big they are to control the selectivity. So it's a completely different set of products than one gets from halogenation or nitration of an aromatic from introductory organic chemistry. So heteroaromatic substrates have their own selectivities, which we don't quite understand, so I won't go into that. All right, so what use does that reaction have then? Right, again, those boron derivatives do a lot of different reactions. And so the concept is by having this one CH activation reaction, which is sterically controlled, that we can then take that to this whole series of products. Now, in a one-pot reaction, you do the first reaction. You don't have to purify anything. You just add another reagent, and you make this whole set of derivatives with now a high selectivity that's controlled by the sterics of that molecule. So one of my favorite reactions is this halogenation. So let me just focus on that one as one of the examples that's published. And so in this reaction, you start with an arene. You do the iridium catalyzed reaction, and then you just add copper bromide, and you make the aryl bromide. And you make that aryl bromide, again, sterically controlled, not electronically controlled like the halogenation that some of you might have learned some years ago. OK. All right, so and again, is this green chemistry, right? So you've got the boron derivative. You come in with stoichiometric copper. And so the key is to think about how else you would make those molecules. It's not that you can't make them. Right? They can be made, but by pretty ugly chemistry. So I turned to Wenyoung Chen in my group to ask him, how would you how much bromide is in there? Wenyoung knows everything. He's a postdoc. So he said he found this example with gallium trichloride as a Lewis acid, and you get about 20% of this material amongst all of these others. And so if that's the product you want, then you'd have to purify this away. Right? But from the chemistry I just showed you, that's the only product that gets formed. An alternative, if you had any starting material, would be to go through this 5-step sequence to get from a hydrocarbon building block like mesitylene to get to the aryl bromide. And so instead of doing these 5 steps, some of them with nitric acid and thionyl chloride and so forth, instead of those reagents, we do our one-pot synthesis to to go from here straight to there. So I think a lot of this is all about the number of steps, number of separations, number of purifications in the middle, and being able to cut all that out through doing catalysis. And so the last reaction I'll talk about then is one that's unpublished, which has sort of always been on the list of some of the brownest reactions, if that's the opposite of green, right? So which is Friedelcrafts chemistry. It has a stoichiometric amount of byproduct. And it has selectivities that, again, are difficult to overcome because of the mechanism and the rearrangements of carbocations that are involved. So if you want to start with an arene and make an alkyl arene, and you want to do that to put a straight alkyl chain on that, it doesn't work very well if this is an electron-poor arene. You get mixtures of primary and secondary products because of rearrangements of carbocations, and then you've got this orthopara versus meta-selectivity of sophomore organic chemistry coming up again. And so those are some of the limitations that I list here. So the question then is, could we use a combination of this CH activation chemistry and then a new version of Suzuki cross-coupling chemistry to be able to do the equivalent of a Friedelcrafts reaction without all of these problems and separations that are required? And so one of the problems then is trying to be able to take the aryl boron derivative we make from the CH activation chemistry and be able to attach that to this alkyl chain. And so that's been a challenge in the area of cross-coupling. Greg Fu's group at MIT has really overcome a lot of those challenges. But still, this reaction was one that was not known for these reasons down here. And so sort of one result, detailed results slides for the talk. So Dan Robbins, a current graduate student who's finishing up, has found conditions by going through different ligands with different nickel precursors that allows for this transformation to be done. Okay? And so by the discovery that L4, this diamine ligand on nickel, will catalyze that reaction, then allows for this process, where one starts with a disubstituted arene. We do the CH activation with the boron derivative to put boron in that position. And now we can couple to put a straight alkyl chain on the aromatic ring. And we can put that alkyl chain on the aromatic ring in a position that can't be put on the ring through Friedelcrafts alkylation because of this orthopara versus meta-selectivity relative to the steric selectivity we see here. And so here's just a few examples of those reactions. It's not a perfect reaction. The yields aren't great at this point. But it's, I think, a really large step in the right direction. And for those of you who are synthetic chemists, we can also do that with secondary alkyl halides. And the hope is that we can do that with a secondary alkyl halide to install a secondary alkyl group without rearrangements where you would get different secondary alkyl attached. So this is sort of a cheating example, but that's a glimpse into what I think we'll be able to do. All right, so the last thing then is just to give you a couple minutes on the mechanism of this and one last reaction that we've been able to develop through that. Okay, so the active catalyst in these CH activation reactions is a compound with metal boron bonds. And there's 3 metal boron bonds, 3 of these so-called boryl groups attached, and then an olefin, which comes off the metal to generate the reactive intermediate. The turnover limiting step, or the slow step of the reaction, is the cleavage of the CH bond by interaction of that CH bond to that position of the metal complex. So we wanted to understand what the properties were of the different groups bound to the metal and how they control the rate of that reaction. If we could understand that, maybe we could make a better catalyst that could do new things. And so if we compare the reactivity of the compound with alkyl groups at oxygen versus an aryl group at oxygen, I wouldn't have thought there'd be such a big difference. But there is. This compound reacts room temperature in minutes. This compound reacts in hours at elevated temperatures. Right? By just changing the group that's on the oxygen, that's on the boron, that's on the metal. So why does that change anything? Well, it changes the amount of electron density at the metal center, how electron-rich or electron-poor the metal is. We can measure that by CO stretching frequencies. The value is given here. And the bottom line is that the more electron-rich the metal center, the faster the cleavage of that CH bond. And so if we want to make a better catalyst, we have to make the metal more electron-rich. So how do we do that? So without going through all the things that we tried and all of our analysis, let me just point out that the original catalyst that we had with the bipyridine ligand gave essentially no product for cleavage of an alkyl CH bond. But if we make a fairly subtle change by putting more substituents on that ligand, and we can do that by switching to phenanthrene instead of bipyridine, and we put this one with more electron donors on the ligand, we can now get good yields and still maintain that very high selectivity for functionalization of a primary CH bond. nobody's going to use this reaction. Right? This is a neat alkane. It makes a product that you could take to an alcohol, but there are other ways to make those linear alcohols. Right? So is there a way, though, to use this reaction in another context? And that's by some work of Eric Simmons, who is a postdoc in my group, actually a PhD student here at Berkeley with Richmond Sarpong. And so what Eric showed is that we could use silicon reagents instead of boron reagents if this was intramolecular. So you start with the alcohol, put silicon on the alcohol. This is a known reaction. Put the silicon on here, and use a silane so that you would leave one SiH bond left. That hydrogen atom comes together with that hydrogen atom at the end of the chain due to the action of that catalyst, drives off hydrogen, and makes this ring system. And so the reason why that's useful is because you could take that ring system and open it up by adding an oxidant and the Tamau-Fleming oxidation to generate a 1,3-diol. Okay? And so we've taken that CH bond at the end of the chain, and because of the position of that alcohol and the way Eric designed that reaction, we can oxidize that CH bond and put an alcohol functional group at the end. Okay, we can also do that on a secondary alcohol without oxidizing the secondary alcohol to the ketone. And so what that allows one to do is to take a relatively complex molecule like this one, which has lots of CH bonds in it, has some other functional groups, but one alcohol. You do the first reaction to put silicon on that alcohol. Then you add the iridium catalyst that does the CH activation. It grabs one of the CH bonds of this methyl group, makes that ring, you oxidize it, and in this sequence of steps, all done in one pot, you oxidize that alcohol. And so people have wanted to do that for medicinal chemistry. This is a set of compounds that have a variety of biological activities. And so this has been done before, but this was done before in 10 steps. And we do this in 3 steps, but it's all in one pot. You add one thing, and then you add the other. So in one set of transformations, we get to this product that took 9 purifications in between 10 steps before. And then we can take this compound and do the same transformation on that. And this has been oxidized by whole cell oxidation. To the extent that one of those 2 methyl groups gets oxidized, it's that methyl group, not that one. And so then we generate a new material, which would take many more steps to be able to generate in some other means. So to summarize then, one of the early points is that catalysis really pervades all of green chemistry. And whether it's using different reagents, less hazardous reagents, or just taking a 10-step sequence and making it one step, those all have a big impact on the amount of waste and how, maybe 10 of those 12 principles. What I really would like to focus on is how some of the catalysis that we and others in our community have developed is really can be thought of as step economy. If you're 45 miles south in Stanford, that's a term that came from Paul Wender, and then atom economy came from Stanford as well, from Barry Trost down at Stanford. And if you shorten the number of steps, you certainly use less solvent. You use less solvents in separations and so forth. And so palladium catalyzed amination was an example of that. And then CH activation was an example of how one can make a new set of molecules, or a set of molecules that, again, would take many steps to make. But by changing the selectivities, we can do that in one step. And so I sort of gave you some snapshots of chemistry that's taken years to develop. I tried to mention the names of the people along the way. I didn't mention Huiyan Chen, who did our first catalytic CH activation, and Karen, who made the first observation of that in my group. But I think I mentioned the others. Qilong was the first to use ammonia. He graduated, and then Yang Vo picked up on that and really turned that into a useful method. So thanks to all of them. And again, thanks very much for the invitation and for your attention here today.
So one of the things I was really inspired by at the beginning of today was John Arnold's comment about the holistic nature of green chemistry, and that there are many people involved in doing many different things. But I also think that's one of the challenges, which is that we have to look to speak a common language. And I have to say that's always certainly a challenge for chemists. But I think Professor Hartwig did a wonderful job of really getting across to us sort of the importance of catalysis, CH functionalization, which are some of the most exciting work that I think chemists are doing right now. Given the time constraints, we'll have time for perhaps just one question or maybe 2. But Professor Arnold Hartwig will be available during the coffee session period for answer more questions. Okay.
Professor Hartwig, thank you so much for an inspiring talk. Sorry? Oh, sorry. I'm Audrey Morris from McGill University in Canada. I mean, one of the natural things to think of from all the beautiful things that you showed is how, I mean, what's the next step would be recycling the boron species. And there's been some interesting work done recently by notably Doug Steffen in Canada also, where he's using also boronic species in a completely different context, but he's able to use them as catalysts. So I'm sure you're doing that right now. So can you tell us a bit more about that? How do you recycle the boronic species?
Actually, I have not come up with any great ideas about how to recycle that. We thought very hard about that when we did the first CH activation. Of course, the product you get from the workup of these reactions is just boric acid, which is, I don't know, it's ant poisoning, but, but it's a non-toxic byproduct. The real issue is how you make the boron compounds, and I didn't go through that here for reasons of time. And it's, there's several steps to get from boric acid back to the reduced boron species. I don't have any great way to do that. Yeah.
Dr. Hartwig, Roger McFadden, senior scientist at Staples. I'm wondering if there's a danger that we are just identifying a more efficient way to create a toxic material in some cases, and where is toxicity considered in this process? Or is it at all considered at the beginning of these stages of this research, or is it done later? At what point?
So I guess that comes down to identifying the targets for the reactions that one wants to develop, right? And I guess principle number 10 is to pick the products that would be safe, non-toxic, and would have a favorable end use, or whatever. What's the term that we used in the first talk? So we're a group that develops the ways that one makes those compounds. We don't choose those compounds. I mean, maybe in the future our research might go in that direction. But the idea is to develop reactions like the ones I showed you that are general enough that if you envision that this is going to be, whether it's a product that you guys would be interested in or someone in some other arena that would be less toxic, there are now going to be methods to make them. And so one thing that I've learned is that if you invent a reaction that's general and a reaction that is easy to use, and then people will use that to make new things. And so it's the end products are dictated by the invention of new reactions. And so the more reactions we have, the more things that we can make. And then other people make the choices about what, we don't make any commercial products. Nothing I showed you here and what we've done is a commercial product. I hope that answers that question. Okay.
Okay, so again, please join me in thanking Professor Hartwig for